Welcome to the first presentation in the 7th webinar series presented by the International Adsorption Society. The IAS is an international organization dedicated to advancing adsorption as a solution to scientific, engineering, and human welfare challenges through the promotion of adsorption research and education. We thank everyone for attending the webinar today. The webinar series has been an immense success, and the recordings of previous webinars continue to be viewed on YouTube and Bilibili. This is the first webinar of our 7th series, which we will continue to have monthly throughout the year. We intend to continue with a variety of speakers from industry, academia, and other research institutes, as well as PhD students and early career researchers. Announcements regarding the 7th series will be distributed through the IAS mailing list and LinkedIn page. Today's webinar will be given by Dr. Benjamin Claessens at Aix-Marseille University in France. I am Nicholas Wilkins at Svante Technologies Incorporated in Canada. Today's webinar will be moderated by Salar Azar at the University of Liège in Belgium. We are required to remind attendees and future viewers that the views expressed by the speaker, host, or other moderators are not necessarily those of the IAS or the institutions associated with those individuals. We ask that you consider joining the IAS as a regular member if you have not already. Our dues are minimal, only $20 per year, but support the publication of our flagship journal, Adsorption, contribute to travel grants, and workshop seed funding for IAS members and affiliated groups, as well as aid the organization of our triennial conference on the fundamentals of adsorption. Members also receive free access to IAS-supported materials, including our journal, as well as the adsorption database published by Springer Materials. Anyone can follow the International Adsorption Society LinkedIn group for future updates regarding IAS events, webinars, and information about scientific meetings. Please help us expand our YouTube channel by liking this video and subscribing. I'll now hand it over to Salar Azar, who will moderate the YouTube Q&A.
Thank you, Nick. It's my pleasure to welcome Dr. Benjamin Claessens to today's webinar session. Benjamin Claessens is an associate professor at Aix-Marseille University, France, where he works in the Madirel laboratory. Research in his group focuses on understanding the fundamental adsorption mechanisms of gases in microporous materials and the use of porous materials for gas separation processes. Prior to working at the Madirel, he studied his PhD in the lab of Professor Yuri Denayer at Vrije Universiteit Brussels, VUB, in Belgium, on the use of hydrophobic adsorbents for the recovery of renewable alcohols from vapor and liquid mixtures. Subsequently, he performed a postdoc at Ghent University in Belgium, where his research focused on the use of ceramic membranes for organic solvent nanofiltration applications. He then moved to Madirel for a postdoc where he studied ion exchange LTA zeolites for alkane-alkene separation. He has been awarded the Ilya Prigogine Prize in 2016 by the International Solvay Institute, a Gustave Buel-Sofina Prize in 2019 for his PhD work, and obtained the 3rd place in the Belgian National 3-minute thesis competition. He is a board member and treasurer of the French-German Adsorption Initiative and junior chair of the 2026 French-German Adsorption Conference. During the webinar, questions can be submitted to the speaker as comments on YouTube live stream, and then questions will be answered at the end of the presentation. With that, I warmly welcome Dr. Benjamin Claessens again and pass over the presentation to them.
All right.
Can everybody hear me and see the screen? Yeah,
Yes.
should be fine. Okay, so thanks a lot for the introduction. And so, as I said before, I'm a huge fan of the webinar series. So it's really a great for me a great honor and a great pleasure to be able to give this presentation today. So I'll be talking about gas adsorption measurements, so coupled to microcalorimetry measurements for the direct measurement of heat of adsorption. So as said in the introduction, I'm a lecturer at the university here in Marseille. And so Marseille, it's a city in the south of France. And so if you start Googling Marseille as your next holiday destination, you'll find out that the city is known for pétanque, which is a typical game which is played in the south of France. It's known for pastis, which is an anise type liquor. And very often people, they do the combination of drinking pastis and then playing pétanque outside. It's a city of many religions, but there's only one real religion, and that's the football team, Olympique Marseille. And it's also the city of one of the most beautiful national parks of France, the Calanque, which gives for great places for hiking and swimming and so on. So that's what you will find if you Google Marseille and if you're looking for TripAdvisor tips. But people working in thermal analysis, they know that Marseille is also known for something else, which is the Tian-Calvet calorimeter, which is a type of calorimeter, so an instrument which measures heat during chemical and physical transformations, which was developed in Marseille in the previous century by Albert Tian and Edouard Calvet. And so what's very specific about this type of calorimeter, which allows to measure heat, is that it's isothermal. So it's not an adiabatic calorimeter. It's not a differential scanning calorimeter, but it's a calorimeter which works at constant temperature, which makes it specifically suitable to study gas adsorption phenomena. And so today I will be talking a bit about this, about how we can determine and measure heats of adsorption. So first of all, to take a step back. So most people working in gas adsorption know this, but gas adsorption is an exothermic process, all right, because a molecule attaching itself to a solid surface, it will lose freedom, it will lose entropy. And this means that there needs to be a heat release to have a spontaneous process. So in gas adsorption, it's a bit like in marriage, all right, you need sufficient attraction to compensate this loss in freedom. So this means that thermal effects, these heat of adsorption related effects, they are important in gas separation processes. So for people doing dynamic column breakthrough experiments, they know this, but this is, for instance, an example which came from a paper by Buzmagian from many years ago, but it shows us the separation of a CO2 ethylene mixture on a breakthrough column. And due to the heats of adsorption, which are released during the adsorption process, the temperature at the output of the column will increase. So in this case, the gas temperature during the adsorption processes increases by about 50 degrees. And this means that if we want to determine equilibrium amount adsorbed from these breakthrough experiments, we have to wait until the full the column is isothermal again. So for people doing breakthrough experiments, they notice that there is this risk of thermal wave formation, but also in cyclic adsorption processes, there is this is a profile which came from a paper by Paul Webley, for instance, working on nitrogen-oxygen separation. And this is a cyclic steady state profile, which was measured in an adsorption column, where at a certain point in the adsorption column, the temperatures decrease up to -10 degrees. So in cyclic adsorption processes, cold spots can form, as well as hot spots at cyclic steady state due to these heat of adsorption, which is released during adsorption and which has to be added during the desorption process. And then, of course, in thermal swing processes, so where we use a heat to regenerate our adsorbent, we need to add heat. And this will be related to the heat of adsorption. So there is a practical interest in determining or knowing heats of adsorption, but there's also a fundamental interest in the sense that measuring the heats of adsorption or determining heats of adsorption can give an idea on the adsorption mechanism of gases inside a porous material. So if we look at the interaction energy of a probe molecule as a function of the amount adsorbed, well, then if we have a homogeneous adsorbent, then this interaction energy will be more or less constant with the amount adsorbed. But if we have adsorbent interactions with a heterogeneous material, where we have, for instance, interaction sites with different interaction strengths, then typically this interaction energy will decrease with increasing amount adsorbed. And then if we think about porous, microporous materials, where there's not a lot of place to put a lot of molecules inside a pores, then adsorbate interactions will become more important as loading increases. And so typically the heat of adsorption is the sum of these different types of interactions, and by measuring or by determining heats of adsorption as a function of loading, we can get some insights into these different processes. So the most common way to determine adsorption enthalpies or heats of adsorption is using the Clausius-Clapeyron equation. So many people will be familiar with this method, but it allows to determine the differential enthalpy of adsorption. It's the equivalence of the Clausius-Clapeyron equation for vapor-liquid equilibria. And what we need in this case is values of temperature and pressure at a fixed amount adsorbed. So the heat of adsorption will be related to the temperature and the pressure at a fixed loading. And so the assumptions which are behind this relationship is that the adsorbed phase is much more dense than the gas phase, that the gas phase behaves as an ideal gas, and that the enthalpy is independent of temperature. But so to apply this relationship, we need temperature and pressure points at a fixed loading, while what we typically measure are adsorption isotherms. So before I forget, the enthalpy, which we determine this way, it's what we call a differential enthalpy of adsorption. So it's the difference between the differential enthalpy of the adsorbed phase and the differential enthalpy of the gas phase. Okay, so typically what we measure is we don't measure temperature and pressure points at a fixed amount adsorbed, but we measure adsorption isotherms. So we will measure the amount adsorbed at a fixed pressure, and then we'll have to recalculate these isotherms to be able to determine the enthalpies of adsorption. So typically what we'll do is that we'll measure adsorption isotherms at different temperatures. So what's very often as a guideline is given is to have isotherms with about 10 degrees difference. And then at a fixed amount adsorbed, if I use these measured isotherms to determine the equilibrium pressures which correspond to the amount adsorbed at different temperatures. And then what we'll do is make these isosteer plots, all right? So we use the Clausius-Clapeyron equation, which tells us that if we plot the logarithm of the pressure as a function of 1 over the temperature, we will find a straight line where the slope of this straight line should correspond to the isosteric enthalpy of adsorption. And by doing this for different amount adsorbed, for different loadings, we can then have an idea of the change of the adsorption enthalpy as a function of loading. So there are some disadvantages in this method in the sense that it's an indirect method. So we don't measure adsorption isosteers, we measure adsorption isotherms. So we have to use these isotherms to back calculate the adsorption enthalpy. So we don't have a direct way of determining them. And then secondly, as I said, we have to interpolate adsorption isotherms to back calculate the isosteric heats. And so the way we interpolate our isotherms can have an influence on the enthalpies that we determine. So this is a paper which came out by a former PhD student in our lab, Amin, this year, who actually compared different, so he fitted adsorption isotherms at different temperatures using different models, and compared these different models for the determination of the enthalpies of adsorption. And depending on the type of material and the isotherms and so on, sometimes they had quite large differences between these the different methods he used to interpolate his isotherms. So it's a method which is very often used, which has its limitations. And so we in our lab, we have some in-house developed systems which allow us to directly measure heats of adsorption during gas adsorption processes. So this is a system and a series of apparatuses which were originally developed by Jean Rouquayrol many years ago, which are based on the type of calorimeter which I showed on the second slide of my talk today. So here you can see inside this big barrel, we have a Thion-Calvé calorimeter, so a calorimeter which is kept at a constant temperature. In the inside, we will have a sample cell and a reference cell. And the sample cell is completely, I would say, covered. There's a series of thermopiles around it, a series of thermocouples, which will measure the heat which is released during an adsorption process. So here you can see a picture, for instance, of a thermopile. So the adsorption cell will go inside this cylinder, and these are the thermocouples which are wound around the adsorbent cell. So you can see another schematic here on the left. So basically, we'll have a sample cell. We will have a long tube where we will dose gas through this tube. Our adsorbent sample will be at the bottom of the sample cell. We have a thermopile, so this series of thermocouples which is wound around all the sample cell. And then this whole system is kept isothermally, so that's why it's in this big barrel, which is kept at constant temperature, so that as we dose gas, after the system is completely at equilibrium, it stays at the same temperature. So since it's an isothermal system, by dosing gas, we can measure the heat which is released and simultaneously measure the adsorption isotherm. So as I said, this is a picture of one of the systems which we have in the lab. So here on the right, besides the isothermal microcalorimeter, which allows to measure the heats of adsorption, we have a second system on top, which is actually equivalent to a commercial volumetric system to measure adsorption isotherms. So it's a gas dosing system with a fixed reference volume, a pressure transducer, which we keep at constant temperature. We can dose gas into this reference volume. We can put a vacuum, so we can control the pressure inside this reference volume, and we can dose gas towards the sample cell. And so by dosing gas, we can calculate quantities adsorbed, and the thermopile, which is inside the microcalorimeter, will give us a heat signal which can measure the heats which are released. So here you can see an example of what we measure. So we will have a certain pressure at equilibrium in the sample cell at the end of a previous dose. We will increase the pressure inside the dosing volume to dose a certain amount of gas. As we dose gas, pressure will decrease. The sample will absorb a certain amount of gas, and the pressure difference between the pressure in the dosing volume and the pressure at equilibrium will allow us to calculate the amount adsorbed, just as in a commercial volumetric system. But then besides that, we'll measure a calorimetric signal. So as adsorption takes place, there will be a heat peak, certain amount of heat which is released, and integration of this heat peak will allow us to determine the heat of adsorption. So to be completely correct, or to give a full overview of this work, of how this works, we have to write a small energy balance over the sample cell. So we have our adsorbent inside our sample cell. We have a thermopile which is wound around the adsorbent cell. We can dose gas into the sample cell. So this means that the internal energy of the sample cell will change as we dose gas. This change in internal energy will be equal to the small amount of heat which is released during adsorption. There's a certain amount of work which we have to do to introduce gas into the sample cell. And the gas which we dose into the cell has a certain amount of internal energy as well in the beginning. The work term, there are two terms inside this work term, one which is related to the gas which will be adsorbed, and then one which is related to the amount of volumetric work, let's say, which we have to do to introduce gas into the sample cell, which is related to VC, where we take into account only the volume of the sample cell which is in contact with the thermopile. So there is a part of the sample cell which is connected to the gas dosing volume, which counts as that volume. But heat effects in this part of the volume won't be measured because we only measure the heat effects for the part of the sample cell which is covered with the thermopile. So we only take into account this volume. So combining these two equations allows us to back calculate the enthalpy of adsorption, the differential enthalpy of adsorption. So this will be equal to the change in internal energy, or the change in enthalpy for the adsorbed phase, minus the differential enthalpy of the gas phase. So this comes back to this isosteric enthalpy of adsorption. And this will be equal to the heat signal which we measure. So the measured heat signal divided by the quantity adsorbed, plus a certain term, which is a correction term which we have to apply to take into account the work which is done to introduce gas into the sample cell. And so the assumption which we make is that the system is reversible, so that it's fully thermodynamically reversible, and that the doses which we do are small enough to get the differential enthalpies of adsorption, so the derivative of the enthalpy for the amount adsorbed. So this means that we have to determine this VC, the volume which is seen by the calorimeter, and this typically we do using blank experiments. So as it's a volumetric apparatus, we have to do a helium blank experiment to determine a dead volume to calculate for adsorption isotherms. So from the same system, we will have a blank heat peak which is released from the calorimeter, and we use this to back calculate the adsorption enthalpies and to know the volume which is in contact with the thermopile. And then this differential enthalpy of adsorption is equivalent to the one which is determined via the isosteric method, but it's measured directly instead of being back calculated from the adsorption isotherm. So this is to give you a bit, an overview of how to determine heats of adsorption and how we can measure them directly. So to give you an idea on how this direct measurement system works. So in the following part of my talk, I will try to give some examples to give you a bit of a feeling with the measurement technique, to give you some examples of what we can learn from this method and so on. So the first example which I'll be discussing was work which was part of my postdoc actually, so which was focused on propane-propylene separation. So why did we look into these molecules very quickly? Because they have a very similar boiling point, very similar kinetic diameter. So in general, alkene-alkene separations is not so easy. And in this specific project, we looked into LTA zeolites. So LTA zeolites are gauge and window type zeolites. So this means that they have a very small pore opening, a small pore opening of about 4 angstrom. Behind this small pore, there's a larger cage of about 11 angstrom. And so since this is a zeolite, depending on the amount of alumina in the framework, there has to be extra framework cations to balance the negative charge which is present in the framework. So these cations can be present on different sites, either inside the pore opening, or there are 2 different sites which are inside the cages. And for an LTA zeolite with a silicon to aluminum ratio of 1, that would be about 12 monovalent cations per LTA cage. So just to give you an idea. So in the examples which I'll show you, or in this research project, we investigated ion exchange LTA zeolites, so zeolites which were synthesized using sodium as a starting form, so with 12 sodium cations per cage, more or less. And then our collaborators in Mulhouse, they performed an ion exchange, so they with calcium, zinc, and magnesium, with either an exchange rate of about 50% or about 25%. So exchanging not all the sodium in the framework, but 50% or 25% by divalent cations. And we also used they also synthesized an all-silica zeolite, an all-silica LTA zeolite, which we used as a reference material. So I'll be showing a bit these results because I think it gives some nice overviews, some nice insights in how heats of adsorption can evolve depending on the type of, depending on the gas adsorbent interaction. So first of all, let's take the example of the zeolite framework, which doesn't contain any cations. So the all-silica LTA zeolite. So here you can see the isotherms which we measured on our in-house device. So you can see the propylene and the propane isotherm. So they more or less have the same initial slope. Okay, in the low pressure region, they are quite coinciding. At higher pressures, there's a small difference due to molecular packing differences. So propylene is slightly smaller than propane. So we will be able to put a bit more propylene inside the micropores of the LTA zeolites than propane. So here you can see then the adsorption enthalpies, which we measure in the same measurement. So the between propane and propylene, there's not much difference in adsorption enthalpies. But what we do see is that at low loading, there are not really strong interactions. We can then see that the adsorption enthalpy increases with the amount adsorbed. So these intermolecular interactions, which I talked about, they make that the adsorption enthalpy increases from about 30 kilojoules per mole to about 35, for instance, for propylene. And then as the pores of our zeolite completely fill up, we will see that the adsorption enthalpy drops off as the cages of the LTA zeolite completely fill. So if we look into the calcium exchange samples, so this was a zeolite containing sodium ions. Originally, we exchanged 50% of the sodium ions for calcium ions. You can see that now there is a difference in the adsorption isotherm between propane and propylene. So in the low pressure region, we can see that the isotherm for propylene shifts to the left. So this indicates a higher affinity for propylene, where there's a double bond in the molecule to interact with the extra framework cations. And if we look into the adsorption enthalpies, we can also see a difference. For propylene, we see this initial increase. We also see it a little bit for propane at low loading, so due to interactions of the prop molecule with the calcium ions. And then as the pores fill up, the enthalpy slowly decreases with loading until it completely drops off when the zeolite micropores are filled. So for propane, this is much less profound. So it follows much more a profile which is typical for a homogeneous prop molecule. So if we now look at propane adsorption, but on these different on LTA zeolites exchange with different divalent cations. We see here in green again the adsorption isotherm for pure silica LTA, the propane isotherm, and then the different divalent cations here. So as there are divalent cations or cations present in the structure, we see that the isotherm shifts to the left. So there's a clear influence of the electric field on the affinity for propane, even though it's an inert prop molecule. But between the different cations, there is not so much difference in the low pressure region. If we look at the adsorption enthalpy profile, so we can see that the presence of extra framework cations leads to an increase in adsorption enthalpy of about 20 kilojoules per mole compared to the pure silica LTA. And then between the zinc, magnesium, or calcium exchange cations, there's not so much difference in the overall adsorption enthalpies. There is this very small increase at initial loading, and then as the pores fill up, the adsorption enthalpy stays more or less constant, increases a bit with loading due to intermolecular interactions, and then they drop off completely as the cages of our LTA zeolites completely fill up. So this is for propane, where there's only the possibility to have weak interactions, van der Waals interactions with the framework. Now let's look into propylene, where we have a double bond inside in the prop molecule. So here again in green, you can see the isotherm for pure silica LTA. And on the left, you can see the isotherm for a calcium exchange LTA zeolite, and then a magnesium and zinc exchange zeolite. So here I put the isotherm in a number of molecules which are exchanged per cage, per LTA cage, instead of millimoles per gram. And what we see is actually that we have two steps in the isotherm for the zinc and magnesium exchange samples. So for the magnesium exchange samples, at a first step, a one molecule per cage adsorption. For the zinc exchanged sample, a first step at 2 molecules per cage adsorbed, and then there is a small inflection before the zeolite fills up. And we can actually see these inflection points reflected in the adsorption enthalpy. So here you can see again in green, the propylene adsorption enthalpy is on pure silica LTA. on calcium LTA. So this is a profile that's typical for a heterogeneous adsorbent. So an increase in adsorption enthalpy at low loading, and then it drops off. And then for zinc and magnesium, we see this first plateau, let's say in the adsorption enthalpies with loading, and then at 1 or 2 molecules per kg, the adsorption enthalpy drops off and it falls, coincides almost completely with the calcium LTA sample. So there's this first step, which indicates a very, very strong interaction with the zinc or magnesium exchange samples. So we can see this step reflected in the adsorption enthalpies. Now, what happens if we looked into the samples which had half the cation content, so not exchange at 50%, but exchange at 25%, we see that, interestingly, for the zinc sample, this inflection point shifts. So as we halve the amount of cations in the LTA sample, we can see that the inflection points also gets, the point where the inflection happens also is divided by 2, so from 2 molecules per kg to 1 molecule per kg. Interestingly enough, for the magnesium sample, this is not the case. So currently we're working together with people doing molecular simulations to actually try and understand what the reason is. But one of the possibilities is that that there's a difference in coordination, so that the propylene molecule doesn't coordinate the same way with the magnesium cation and with the zinc cation. But there is also the possibility there are some steric effects playing a role. And so what's interesting is that if we compare with pure sodium LTA, this second step, plateau in the adsorption enthalpies, seems to be independent of the type of cation and coincide with the one for sodium LTA. So I hope I've been able to show you that indeed these microcalorimetric experiments, they can give some insights into the mechanisms of adsorption in these type of zeolites, and also that the type of extra framework cation matters. For propane, matters much less than for propylene. And that, for the zinc and magnesium exchange samples, we have a complex two-step adsorption mechanism, where the measurement of adsorption enthalpies allow us to have an idea of the interaction strength with each different site. So that's just to give you very briefly, to give you an idea of what kind of results can come out of these this measurement technique. So as I told you in the very beginning of my talk, the adsorption enthalpies which we measure are essentially equivalent to those which you determine using the isosteric method. And so this led one of our PhD students, so Amin, who left the lab a few years ago, to the idea, well, can we not do the inverse thing? If we measure an isotherm and if we measure the adsorption enthalpies, can we not predict adsorption isotherms at different temperatures by rearranging the Clausius-Clapeyron equation? And so turned out that this worked. So his paper came out this year, actually. So he measured CO2 isotherms on different kinds of samples at 30 degrees, and you can see some of the results here. So the green curve is the isotherm which was measured using adsorption manometry coupled to microcalorimetry, and the other symbols are isotherms which are measured, if I remember correctly, on a different apparatus, on a commercial apparatus, and the dashed lines are predicted isotherms. So based on this measurement, measured at 30 degrees, together with the adsorption enthalpies, he was able to predict the isotherms at different temperatures quite nicely by inverting the isosteric method. So another example which I wanted to briefly discuss was work which was done by not by me, but by my colleague Sandrine in the lab, and Philippe when he was still in the lab. They worked a lot on metal organic frameworks and understanding flexibility in metal organic frameworks. So the results which I will show very briefly afterwards are on the MIL53 adsorbent, which were obtained during the PhD of Sandrine, if I remember correctly. So the MIL53 MOF, it's one of those classical examples of a breeding MOF. So it contains one-dimensional, it's a one-dimensional pore system. You have metal octahedra, which are connected by benzene dicarboxylate groups. And so this material can open and close a bit depending on the gas molecules. And so Sandrine And Philip, what they measured was, so here you can see an adsorption isotherm of methane and CO2 on this material. And so for methane, in fact, when the MIL53, when it's activated and dehydrated, it's in its open pore form. And so methane will adsorb in this open pore form according to a Langmuir type mechanism. So it's a type 1 isotherm. While for CO2, there are distinct steps in the isotherm and a hysteresis loop, which is very typical. And this is related to the framework closing and then opening again at higher pressures. And what's interesting is that this gives adsorption enthalpy profiles which are very typical, which are quite exotic. So for methane, we get a profile which is similar to let's say a neutral probe molecule adsorbing in a homogeneous energetically homogeneous material. But for CO2, this is not the case. So we can see these two peaks in the adsorption enthalpy profile, which is related to CO2 adsorption in the closed pore form. So as CO2 adsorbs, the framework will collapse in its closed pore form. And then this drop in adsorption enthalpy, where the adsorption enthalpy even drops below the vaporization enthalpy, is due to opening of the framework. So as the framework opens, as gas adsorption takes place, a certain amount of energy is needed to open up the framework, and this is reflected by this sudden drop in the heat which we measure, which is then followed by adsorption of CO2 in the open pore form. So using microcalorimetry, we can follow these different steps in opening of the metal organic framework. And so they test a lot of probe molecules on this material, so methane, oxygen, argon, nitrogen, where they all observed a more or less homogeneous profile. So the adsorption enthalpy being relative, showing profile which is typical for a homogeneous adsorbent. What's interesting is that with linear alkanes, so when testing methane, ethane, propane, and butane, we would expect these linear alkanes to be also quite inert. But in fact, in the adsorption enthalpy profile, there was a peak which was present. So for ethane, propane, and butane, which is not present for methane. So even though there is no really, so CO2 as a quadrupole moment, so there is a possibility for a strong interaction of CO2 with the MIL53 adsorbent. But for these molecules which are more inert, was not something really to expect, but there appears to be also a narrow pore transition for these molecules. And so this led my colleagues at the time to propose a certain minimum amount of energy, minimum interaction energy, which is needed to be able to open and close this framework. So what they saw looking at these different probe molecules is that as the interaction energy, so the zero loading enthalpy, is below 20 kilojoules per mole, there is no opening and closing of the framework observed. While as molecules become larger, or the adsorption enthalpy at low loading increasing, the enough energy is provided to open and close them off. So this is one example on more flexibility. And then a final thing which I wanted to talk about, because it's a very cool application. This was developed by Jean Roquerolles many years ago, but it's a cool device. It allows to measure heats of adsorption at cryogenic temperature. So the idea was to have a kind of a technique complementary to the measurements of nitrogen or argon at cryogenic temperatures, which you use to determine pore size distributions and so on. Where Jean, in fact, wanted to put, so you can see here a picture, the microcalorimeter inside a vessel of liquid nitrogen. So the idea is quite crazy. So they designed this system actually as a diving bell. So they put the microcalorimeter upside down to avoid that there's direct contact with the thermocouples and the liquid nitrogen or the liquid argon. And there's a flow of helium which assures that there is no direct contact of liquid with the calorimeter, and then the sample cell is a U-shaped tube which is in contact with the calorimeter. So the idea behind these measurements, and another special thing about these measurements is that the dosing is no longer discontinuous, but they developed a continuous kind of dosing. So using a sonic nozzle, so dosing a continuous flux of gas, actually a fixed flux of gas, which allows to directly determine the differential enthalpies of adsorption by following the heat signal of the calorimeter as a function of time and the pressure signal of the pressure transducer as a function of time. And in fact, this is a method which is tricky in the flat parts of the isotherm, in the plateau part of the isotherm, because dead volumes will be very important. But in parts where the isotherm is very steep, the amount of gas which we dose will essentially be completely absorbed. And so this is a region of the isotherm where this method works very well. So I will show some examples which were again provided to me by one of my colleagues, which was on the MIL 100 chromium MOF, which is a MOF which contains two different pore sizes, two different gauges of pores. So smaller cages of 25 angstrom and larger cages of 29 angstrom, which each had different opening sides, and also uncoordinated metal sides. So this is on the chromium one, so there were some chromium ions not coordinated. So here you can see some of these isotherms which are measured. So for carbon monoxide, methane, and nitrogen, so at 77 K. So by putting the calorimeter in a vessel of liquid nitrogen. And so in these isotherms, you can see different steps in uptake. And so here you can see the differential enthalpy of adsorption on the right. So you can see the difference for carbon monoxide and nitrogen, where you have at low loading a very strong interaction with the uncoordinated sides. Well, for methane, this is much less profound. Then there is a plateau which corresponds to filling up of the cages. So adsorption on the internal surface of the cages. And then as the different cages fill up, there's a small peak in the adsorption enthalpy profile, which corresponds to the complete pore filling of each cage. Voilà, so this gives some this gives an example of this these kind of measurements, which are kind of complementary to gas measurements at cryogenic temperatures. So, so to summarize a bit my talk, so I hope that I've been able to show you that by coupling gas adsorption measurements to these microcalorimetric measurements, it gives extra insight in adsorption mechanisms. It allows to get an idea of the interaction energy with certain adsorption sites and so on. We can also use it to predict isotherms at different temperatures. So inverting the isosteric method, we can predict isotherms at different temperatures than the measurement temperatures. And by performing these microcalorimetric experiments at 77 Kelvin or 87 Kelvin, so at cryogenic temperatures, we get some information which is complementary to the kind of measurements which we do to determine BET surface areas and pore size distributions and so on. So then I would like to thank, of course, my collaborators, so Sandrine and Emily, for giving me some other examples to show as an example of applications of this technique. Jean Rouquayrol for being Jean, and then Amin, who was a PhD student, a previous PhD student in the lab, but I used some of his results from some of his papers as well today. The people in Mulhouse who prepared the LTA zeolites, Michael Badawi, who is performing molecular simulations, so to better understand the adsorption mechanisms of alkenes in these LTA zeolites, and then the people where the lab collaborated with previously regarding MOF synthesis, Guillaume Morin, Montpellier, and then the people in Caen for spectroscopy analysis. And then that will be the end of my talk. So if you want to know more, there's quite a good description on adsorption thermodynamics and this measurement technique in the book by Jean Rouquayrol. There's a paper from Philippe Léoline from already a few years ago, but where he also explains the principle of this measurement technique and gives some example, but more related to zeolite materials. If you want to know more about adsorption thermodynamics, so there's this paper from the Adsorption Journal by Alan Myers and Peter Monsons, which I like a lot, from 2014, which gives a nice overview of the definition of adsorption energies, enthalpies, and so on. And for the connection of, let's say, adsorption energy balances in fixed bed adsorption processes, there's a series of papers by Doug Levin and Walton in the early 2000s as well, which are a very, very good reference. And so if you want to know more about this technique, about these methods, or if you have any questions afterwards, don't hesitate to contact me, and I'll leave it there.
Thank you so much, Benjamin, for your insightful presentation. We have some questions from our audience that I'm going to ask you. The first one is, are there differences or difficulties linked to measurement of heat of adsorption, measurement of chemical adsorbents compared to physisorbents? Is it also possible to somehow measure that using this microcalorimetry method?
I think so. If I remember correctly, it has been used. In our lab, we never did it because there are other catalysis labs in France. I'm thinking about Lyon in particular, where if I'm correct, they use these kind of methods also to look into catalysis. But we mainly look into physisorption processes. But I think so.
Okay. And just to follow up on this question, do you think we would see some signals on microcalorimetry if that shows the reaction happened on our chemical adsorbent, or it would need some other kind of, I would say, to make some kind of revamping on the device to be able to see that?
No, I don't think so. If I remember correctly, there are some articles on this. But you measure the bulk heat which is released during the phenomena which is happening inside the sample cell. So then how to exactly extract which data for which reaction step and so on. For instance, I'm not really a specialist to answer those kind of questions. But I guess so. I guess the signal which will measure will be the overall heat of reaction, for instance, I guess. Mm.
Thank you. And for the next question is, how does the Claessens method change if the gases are non-ideal? Is it still acceptable to use this equation, this method, if gases are non-ideal?
A good question. We should look. I think the paper by Myers and Monson is not a bad thing to go to answer that question. It has to do with with the reference, not really with the reference state, but in fact, in one of my slides, you will see that at a certain point there's the internal energy of the gas minus RT, and it's this RT term which is related to an ideal gas, which is the enthalpy of an ideal gas. It's its internal energy plus this RT term, which is no longer valid for a non-ideal gas, and so then you have to see how the equations change. So I don't know exactly. You should delve into the equations then to see how this changes. But there will be probably a small correction to do. Yeah, I guess.
Okay, thank you. And we have one extra question from our YouTube viewers. Casimir LP says that, I'm not sure if I understood the blank measurement correction. The pressure change over dosed amount between a blank and a sample, and hence the volume work should be different or not.
Yeah, so in fact, what we do is we put our sample cell inside the calorimeter. We do a blank experiment with helium, which anyway we have to do. If you do adsorption manometry, you have to do a blank experiment with helium to determine the dead volume to be able to calculate the isotherm and to do the dead volume correction when you determine the isotherm. But what we will measure as we dose helium, we consider as for adsorption that helium is a non-adsorbing gas, but we will see a certain heat peak. And this heat peak will be related to gas expanding in the sample cell, where we say, okay, this heat peak is related to the amount of work which we have to do to introduce gas into the sample cell. And then we use this same correction when we actually measure the isotherm.
Okay, got you. And another question is, why might one use or not use numerical derivatives instead of an isotherm fit for Claessens-Clapeyron equation estimations?
I'm not sure I 100% understand that question actually. What do you mean with numerical derivatives?
So instead of using the fitting points or isotherm values, just using the derivatives of the isotherm to fit your Claessens-Clapeyron equation.
It should probably work similarly, right? You have to, in a certain way, determine the equilibrium pressures and temperatures which correspond to a certain amount adsorbed. So it's instead of either you fit to use a curve fitting tool or you interpolate directly.
Because if I understood correctly, these fittings they are model independent, right? I mean they should be model independent.
In theory, they should be. But there can be some difficulties also with for instance, determining quantities adsorbed at low loading. So then even if you determine if you interpolate, for instance, directly, could be that you have some more scatter on the isoters when you make the isosteric plot. Ideally, what you want is almost a straight line, but sometimes it depends also on the system. Can be difficult to have good data in the low pressure region, for instance, or have good data in the low pressure region for all temperatures that you want and so on. And so this can lead to some scatter when you make the isosteric plots. So it can be more complicated than it seems at first. Yeah.
Okay. I have to also correct myself because the model itself is dependent. I mean, because it changed the parameters, but the derivative is independent.
Okay.
So we are going to the next question, if I may.
Sure.
What would be the relative uncertainties or error bars in microcalorimetry method compared to Claessens-Clapeyron method or in both?
It's a good question. So this technique, in fact, is best suited in the initial parts of the isotherm. So where the amount of adsorbed is large, and the calorimetry itself is quite sensitive. The difficulty lies in the determination of the amount adsorbed. And so in the initial part of the region, the amount adsorbed is very high. So the error on the determination of the amount adsorbed is smaller. But then as the pores fill up, as you reach a plateau, the delta N, the difference in amount adsorbed between each dose decreases, which means that it becomes more difficult to calculate the enthalpies. Plus, of course, since it's adsorption volumetry, the errors accumulate during the measurement of the isotherm. So the error on the amount adsorbed at the end, and so also the error on the adsorption enthalpy, which is the heat which we measure divided by the amount adsorbed, will not be the same. So it's a method which works the best in the initial parts, and then for parts where the isotherm is more flat and so on, it works a bit less good.
So do you have kind of approximate value for the error for, I mean, in unit of kilojoule per mole, or the order of magnitude, I mean?
From experience, we are at a few kilojoules per mole.
Okay.
Yeah.
Okay.
For instance,
And final questions from our audience is, what modifications are required for the apparatus to ensure condensation does not occur in these experiments?
Okay, complicated. What we do, so we have one system where we do vapor measurements, but they're not very easy. We put we make sure that the calorimeter is the cold point. so that the calorimeter is at the lowest temperature. So I'd say, I don't know, at 30 degrees, for instance, and then we put the
Yeah.
reference volume at 40 degrees. So to make sure that everything else is heated warmer than because what you want is that the vapor adsorbs at the sample or condensates at the sample. So you want the sample to be the coldest point, but it's not so easy.
Okay. Thank you so much for your answers. And thank you for your presentation. I'm going to hand it over Nick for the closure of the webinar. Thank you so much.
Perfect, thank you.
Thank you, Dr. Benjamin Claessens, for your presentation, gas adsorption coupled to microcalorimetry for direct measurement of heats of adsorption. We also thank all of our attendees for joining this webinar, and we hope that it was both educational and enjoyable. An edited version of this webinar will be posted on the IAS YouTube channel and Bilibili. The hope is that the work discussed here today will be a useful resource for the adsorption science community in the future. The next confirmed IAS webinar will be given by Dr. Yurian Boone on March 25th at 1400 UTC. We are also restarting our webinar series in time zones better suited to those living in Asia and the Pacific. The next confirmed IAS webinar in this series will be given by PhD student Hassan Muhammad on April 17th at 0200 UTC. Announcements regarding the next webinar and other IAS online events will be posted on our LinkedIn page and through the IAS mailing list. With that, we thank you for joining us and hope you will join us again soon.